Hello friends, I am Ayantika and in this video I am going to discuss about the reproduction in a bryophyte Pellia. Now Pellia, it's a bryophyte, which means that it has got character that is intermediate between the algae and the pteridophyte. The reproduction in the bryophyte, it is dependent upon water because they have got motile male reproductive gamete, and it has got also got characters that are fit to live in land. For example, the rhizoids that help the thallus to be attached with the substratum as well as for water absorption. So we see that the bryophytes, they are have got adaptation of both aquatic and land dwelling plants. So they are the intermediate, they form the ecotone species between aquatic and the land dwelling plant. So this video I am going to discuss about the reproduction in Pellia. Earlier I've made a video on the overview of Pellia, where I discussed the thallus structure and its ecological importance and general characteristic. So before watching this video, that is reproduction, if you don't have any idea about what it is, so you can go and watch that video, that is the Pellia species overview first, and then come to this video. Here I'll be discussing the sexual as well as the asexual reproduction and the life cycle of Pellia species. So, vegetative reproduction in Pellia occurs by three processes. One is the adventitious branches. We know that any branch which are out of place are called adventitious branches. So, these type of in Pellia species, usually they have a dichotomous branching, but any branches that do not occur at the tip, at the apex of the growing point, the usual position, they are termed as adventitious branches. Here we see that adventitious branches, this is the dichotomy, and each of these are the lobes. So this adventitious branches, they develop from the upper portion. This is the adventitious branches that it is develops, that which means that adventitious branches in Pellia, they develop from the upper surface of the thallus. They develop from the upper surface of the thallus. Gradually they get detached and develop into a new plant. So this is the adventitious branches, how it propagates. Then there is the fragmentation. We know that in case of the bryophyte, if this is the thallus, for example, then the cells in the posterior region, these cells, they gradually get old, they die, and when this death of the cells reaches this dichotomous region, then each of these lobes of the thallus, they form a separate plant, and they again in turn develop dichotomy and develop into a complete new plant. So this is the fragmentation. Then we have regeneration. We know that in bryophytes, they have got a very important characteristic of persistent apex. But here any portion of the thallus, any portion, not just the apical region, but any portion, it can be a ventral dorsal region, it can be a posterior region or the apical region, any portion of the thallus can, if gets fragmented, it can generate a new plant. So this is the regeneration from any part of the thallus. So these are the vegetative reproductive strategies, and these vegetative reproductive strategies, it usually occur during the when food is abundant, water is abundant, which means that vegetative reproduction occur during the rainy season, and at the end of the rainy season, that is when the late rainy season or the spring season, the sexual reproduction occur. And sexual reproduction, it is a chance event, because in Pellia some species are monoecious, some are dioecious. For the dioecious species, where the sex organs are born on different sex organ born on different plant, so for the sexual reproduction in dioecious species, the presence of opposite sex plants in the near vicinity is very essential for the sexual reproduction to be very successful. That is why vegetative reproduction is a very common method of reproduction in Pellia species of bryophyte. So, the reproduction in Pellia species, just like other bryophytes, it is oogamous. Oogamous means that the sex gametes, the gametes are of different both physiologically and morphologically. That is, one of the gamete, that is a male gamete, is much smaller in size and spiral in shape and is motile, that is, it can swim. And on the other hand, the female gamete is much larger in size, and it is circular or ovate in shape, and it does not move, it is non-motile. So this type of reproduction, where the gametes are dissimilar both in shape, size, as well as more physiologically also, this type of reproduction is called oogamous reproduction. That is, we find in this Pellia species. So the reproduction is oogamous, and the male reproductive organ, that is the antheridia produces the sperms, and that is found immersed in the thallus. So if this is the TS of the this is the TS of the thallus, we see that along the midrib region, the antheridia, they are found immersed inside the thallus. They are embedded in the thallus. The antheridia, they are present in the antheridial chamber, and this antheridial chamber, they are found either scattered. See here, they are scattered, but here we see along the midrib region, this is the midrib region, they occur as one or two rows. These structures, here we see, these are the antheridial chamber, and which open to the outside by an ostiole. So these structures are the antheridial chambers, and the archegonia, they are born here under this involucre. In this region, in the apical region, the archegonia, that is the female reproductive organ, are born. But this type of structure, where the female gametangia and the male gametangia, they occur together, this type of species is it is called the dioecious species. That is, the sex organ, they are present. So this is how the TS, the antheridia, they are born. So this is the female antheridial chamber, this region. But in case of the monoecious chamber, they are born. They have narrow opening. This is the ostiole. This is the ostiole, and this is the antheridial chamber, and this structure it is the antheridia. Inner contains the antheridial mother cell, and the outer, this one is the, sorry, this one is these cubical cells, they are the antheridial and androcyte mother cell, and the single layer of sterile cell, these are the jacket cells, or which are sterile, and this single layer of jacket cells, they do not have any participation in the formation of the sperm or androcyte, but the inner cells, that is the antherozoid mother cells, they are responsible for formation of the sperms. Usually the androcyte mother cell, they divide by mitosis to produce each androcyte mother cell. They produce as 2 androcytes, or the spermatids, and these each of these androcytes or the spermatids, they gradually metamorphose to the biflagellate sperms. So the spiral biflagellate sperms of the Pellia, they are produced inside these chambers. So as the sperms, they are matured, and this upper layer of the jacket cell, they imbibe water, they swell, they show hygroscopic movement, and then the upper portion, it gets ruptured, and the sperm that is formed, it swims out through the, and this is how in the entire water that is present on the upper surface of the thallus. Antheridia, so this is how the sperms on the upper surface, liberated the dorsal surface along the midrib. On the upper surface of the thallus, cell can act as an antheridial initial. Any cell close to the midrib region can act as an antheridial initial cell, any superficial cell. So these antheridial initial cell, they enlarge in size and gradually divide into antheridial mother cell, the transverse division into antheridial mother cell and the basal cell. Now basal cell stays as it is. The antheridial mother cell, the upper cell, they undergo further transverse division into primary antheridial cell and the primary stalk cell. Now the primary stalk cell gradually develop into a stalk by division, and the antheridial cell also undergoes division, periclinal, anticlinal divisions, to form the outer layer of wall initial. This outer layer we see, these are the wall initials, and this inner layer of androgonial cells. So this is how this is the longitudinal section. This one, E is the longitudinal section, and in F we see this is how it looks in transverse section, that is the wall initial and the inner androgonial cells. So gradually by further division, the outer wall is formed, and the inner androgonial cell, by repeated division, they form this androcytes, and this at the maturation, the jacket cell tip of these cells, they absorb water, they get the wall ruptures due to hygroscopic movement, and the sperms, this is the sperm that is liberated to the outside. So the female gametangia, it is called the archicomia, dissolving of the outer jacket layer very close to the growing apex, and that is in the terminal portion, that is the apical portion. They are on the upper surface and of the thallus, and they are not born singly, as because the upper surface has got a very thin film and this of water. Cluster, it is protected by a involucre. Earlier, when we have read about the Riccia species, we see that the Archegonia were embedded in the thallus, but and in Markensia, the Archegonia, they were born on stalked, elongated stalk structure called the gametophore. But here in Pellia, we are seeing that they are born very close to the thallus, on the apical region of the thallus, and they are protected by involucre. Now the main function of involucre is to protect the sex organ from desiccation and heat, because we know that bryophytes, this Pellia is a bryophyte, and though it's a bryophyte, but it does not have much protection. That is in the it is not multi-layered. It does not have a cutinized outer wall. The cell does not have this things, so they need some protection against this desiccation. So that protection is provided by this involucre. And note one thing, that earlier when we see in Riccia, Markensia or Lunularia, the Archegonia and as well as the Antheridia, they develop in a acropetal order. But here we see that such order is not maintained, because we see that the central Archegonia, it is quite immature and it is developing. This one is the developing Archegonia, the central one, and the peripheral ones, they are much more mature. So we see this archegonial cluster, and among this archegonial cluster there are also certain multicellular mucilaginous hair. Multicellular mucilaginous hair are also present, which is not shown in this diagram. And the main function is to secrete mucilage, so that this sex organ, they are protected from desiccation. So this is how the female gametangia looks. That is, they have got neck canal cell, and the number of neck canal cell is 6 to 9. These are the neck canal cells, and after the 6 and 9 neck canal cell, there is a single ventral canal cell, and under the ventral canal cell there is an egg cell. So as the gametangia mature, all these neck canal cells as well as this ventral canal cell, all these cells, they degenerate. Usually they degenerate, and the entire, as it degenerates, the entire cell mass, they are converted to mucilaginous
this is
mass.
how the
And this entire neck region, ventral region, it is filled with mucilage
cell
to create easy passage. They enlarge and divide transversely into archegonial mother cell and basal cell. Again, this archegonial mother cell enlarges and divides transversely into the archegonial initial cell and the basal initial cell, the stalk initial cell. And the stalk initial cell later forms a multicellular stalk, and the upper cell, it undergoes further division to form the jacket initials and the ascogonial initial. Now by repeated division, this type of structure, this type of final structure is formed, and we see that this neck and the venter and the egg, they are protected by a single layer of sterile cells that persist even after fertilization as a calyptra. So fertilization, it occurred during the late summer, that is the end of the monsoon season. So what happens is that the archegonia, the archegonia, this is the archegonia with this egg cell, and these are the jacket cells of the archegonia, and by this time the all the neck canal cell and the ventral canal cell, they have disorganized and they converted to mucilage. So the sperm, they swim and enter the neck and ultimately fertilize. So this swimming of the sperm towards the archegonia, towards the egg cell of the archegonia, it is guided by two process. One is the thin film of water, and that helps in the swimming, and another is that the mucilage that is produced inside the female gametangia contains proteins and potassium ion that act as a chemoattractant for the sperm, which guides the direction of swimming of the sperm. So once they are fertilized, so zygote is the ultimate structure that is formed. Then this zygote develops a thick wall and divides repeatedly to form the embryo and later the sporogonium. So zygote, embryo and later the sporogonium, they are retained inside the female gametangia, and the female gametangia also increases in size with the increase in size of the sporophytic generation. So zygote, it is diploid, and it is the first stage of the sporophytic zygote generation. It is the first stage of the sporophytic generation, undergoes repeated division to form embryo, which undergoes repeated division to form a sporogonium, which is a much complex structure. And sporogonium, it consists of three parts. One is the capsule, which is a ovoid or spherical structure and usually green in color, but as it matures it turns black. And the capsule, it consists of spores and other structure called elater. Now the spore, they are haploid, that is they are the beginning of the gametophytic generation or the haploid generation. So the spores are liberated by the dehiscence of the outer wall of the elater of the capsule. They dehisce, and the elaters, they push the spores out of the capsule. So this is the capsule, and capsule has an intermediate region called seta and a foot region, a bulbous foot region called foot. So the main function of this conical foot region is absorption of nutrition from the gametophyte, because the sporophyte in all the bryophytes, the haploid the diploid generation of all the bryophytes, it is nutritionally dependent upon the gametophyte or the haploid generation. So this foot is the sole food absorbing region, nutrition absorbing region, and also helps in the attachment of the sporophyte to the gametophyte. So seta, the function of seta is that as the the capsule, this capsule as it matures, that is the sporogenesis is complete, the intermediate region, that is this region, it elongates and it brings the capsule to a much higher region. Here we see this is the involucre region. We know that the female gametangia, it was born in the involucre region, and the fertilization occurred inside this involucre region. So and the sporogonium also, it developed inside this involucre. But as the capsule matures, this intermediate region called seta, it elongates. This seta is a stalk-like structure, cylindrical structure that elongate, and the capsule that is present at the top of the seta capsule, it is brought to the much higher position. So this elongation of seta helps in dehiscence of the capsule and dispersal of the spore that I'm going to discuss in the next. So this sporogenesis, this is how the longitudinal section, this is the sporogonium. This one is the sporogonium. Here we see this is the triangular foot seta. This one is a seta region, and the capsule, it has got this is the spore, this and these elongated structures, these are the elaters, and these are the basal elaters. So this in Pellia species, we see that the capsule is quite characteristic. That is, the capsule has got not spores, elaters. Two type of elaters. One is the free elater, another is the elaters that are attached to the base. They are attached to the base as bunches. Here we see they are attached to the bases. All the elaters, they are attached to the bases. At least 50 to 100 such basal elaters or elatophore they are present, and the sole function of both the free as well as the basal elater is to help in dispersal of the spore. Now, as the sporogenous mother cell, it undergoes meiosis and rapid division to form the spores, that is the haploid spores. So as the haploid spore, here we see this is the haploid spores. They are this haploid spores, they undergo germination even inside the capsule. That is called the precocoious germination. So at the this maturation level of the capsule occurs, so the intermediate region seta, seta, it elongates, it increases in length, elongates, and the involucre that was covering it, that is ruptured, and at the same time it is brought to the much higher region. And we know that the capsule wall, it is made up of 2 or 3 layers of cells, which is another characteristic, because in Riccia we see, and in Markensia the capsule layer was just single layered, but here we see 2 or 3 layer, and which has got the thickening in the upper region, and also some radial thickening. So these what happens is that as the capsule, it is subjected to arid kind of air, that is the dry air, so hygroscopic movement occurs inside the capsule wall, and the capsule wall, it dehisces. Say this is the capsule, and capsule wall dehisces along along the capsule wall. It dehisces along longitudinally along 4 longitudinal section. Here I have drawn only 2, and other 2 will be available on the other side. So in this way, along the 4 line of dehiscence, the capsule wall will dehisce, and it will expose the mass of spores, elaters, and the basal elatophore. So the spores which has already started germination will be liberated by the elatophore or the elaters, and in this way they will travel to much to some distance, and if they reach a substratum, they will start further germination. Note that one of the characteristic feature of Pellia is that the spores, they start germination even before the capsule is ruptured. So practically the spores are not liberated, germination of but the sporelings inside the capsule, it is called precocoious germination. So this type of germination is also very common in the mangroves. They become multicellular. The mangroves, which are angiospermic plant, their seeds also even before liberation from the mother plant, they have developed into root, stem, and leaf-like structures. So they have there also this type of germlings are liberated rather than the seeds are liberated. So back to the Pellia. This precocoious germination, so the spores, they become multicellular structure, and this multicellular structure, I'll be explaining it in the next slide, that is much elaborate. This is how the spore germinate into a sporeling. While still inside the capsule, we see that it undergoes transverse division at first, and then further division, and this is how, this is a single this one is a single sporeling, and this is another sporeling. This is, they are showing that as the four tetrads are formed from a single spore mother cell, so this tetrad, they do not separate, and each of the spore, they develop into a germling while still in connected. So these are the three spores, one is not shown, and so by repeated division, this rhizoid-like structure is formed, and the lower region forms the rhizoid, the upper region, it forms the apical region, and this is how the final sporeling looks. And while at this stage, that is the K stage or J stage, here we see the spores are liberated, the sporelings are liberated from the capsule, and on reaching the substratum or ground, the Pellia gametophyte is formed. So this one, this is the spore, and this is the sporeling. This one is the final sporeling. This is the spore, oh, sorry, my bad handwriting. So this is the life cycle in Pellia. We see that this is the structure. Is how the it is A, germination, it is of the spore occurs. This one is the gametophyte, which bears gamete. This archegonia, it is here, that develops the male gamete. That this one, C is the antheridia, and B is the archegonia. This is the female reproductive organ, and this is the male reproductive organ. So ultimately these are the structures which are formed, that is the sperm and the egg. So after, this is the process of fertilization, that is after the egg and the sperm mature, F, that is the fertilization occur, and the zygote is formed here, and the embryo is formed by repeated division, and ultimately the sporogonium, this one, the structure, that is the sporogonium, that is the, so we see that here this zygote, it is the beginning of the diploid or the sporophytic phase, and continues till embryo and the sporogonium. But the spore mother cell also, so after the spore mother cell, the spore mother cell undergoes meiosis to form a tetrad, This is a, we see only three um, spores, but another spore is hidden from view. So each of these spore, they form a globosporonema or a sporling, and they are liberated at this sporling stage, and on reaching the ground, young thallus is formed. So this after fertilization, the sporophytic generation starts with the zygote, and then so that was all about the reproduction of Pellia, both vegetative and sexual reproduction and the life cycle of the and I have also made other videos about other type of bryophytes. On the links I have put in the description box. I've also made videos on algae, fungi and lichens, and I haven't put that links here, but you can go to my channel and watch that. And thanks for watching this video. If you like it, do share it with your friends and do subscribe, because your subscription means a lot. Thanks again for watching this video, and next I will be making a video on another type of bryophytes. Thank you.